Please to prove.
All right. Good morning, everyone. Thank you all for coming. I just want to remind you a couple of things. First, please keep your cameras and your microphones off. And we're going to wait for questions until the end. Feel free to either turn on your microphone when the question time begins, or just write it in the chat. It can be either in English or Spanish. We'll be happy to translate. Michelle is also a perfectly fluent Spanish speaker, so she will be able to read it in both languages. And with those instructions for all of us, it is my super great pleasure to introduce Michelle Elise Spicer. Michelle Elise Spicer is a plant community ecologist who works primarily in forest ecosystems. She did her undergraduate degree in an interdisciplinary engineering program from Lehigh University in Pennsylvania. Then she's like, oh, I don't know, maybe being in the field is much fun than being a chemical engineer. And then she did her master's in earth and environmental sciences, also at Lehigh. Then she moved to the University of Pittsburgh, where she did her PhD with Walter Carson. And I'm going to mention something briefly after that about Walt. And then she obtained a very prestigious NSF postdoctoral fellowship. And then she conducted her research at Puget Sound in the West Coast of the United States, and then moved to Yale University, where I had the privilege to overlap when we were both conducting our postdocs. And it's really funny because Walt Carson is Michelle's supervisor, PhD supervisor, and Walt was also the PhD advisor of my PhD supervisor. So we're almost like academically related. Like you're like my aunt or grandpa or grandma. I don't know what that comes to be, but it was amazing to overlap with Michelle during her postdoc. And given that we're both tropical ecologists, we talk a lot about many different topics. And it's really amazing because she has a really broad, but at the same time, very exclusive field of work that I really envy. So her research broadly focuses on understanding three parts of ecology. One is the distribution and drivers of plant diversity across the globe. The second is the ecology and evolution of epiphytic plants, specifically in the canopy, and how natural and anthropogenic disturbances change community dynamics and resilience. So it is my great pleasure to have Michelle with us. And Michelle, please take it away.
Gracias, Sergio. Thank you so much for having me here today. So please let me know if you need me to speak up. I'm a little under the weather today. But like Sergio said, I'm going to be talking a little bit about my canopy work today. So thinking about these drivers of plant community assembly in temperate and in tropical canopy communities. So I really like starting with this photo because even if you haven't spent much time in cloud forests, you can just look at this picture and see that there's such a full and varied plant community just by looking at this picture. And so a sort of central question is that if all of these plants just need a few basic resources, water, light, and nutrients, how can so many of them coexist for long periods of time? And this is a big driver of why we are interested in ecology, why I'm interested in tropical forests. And it's also an important thing to understand since we know that these diverse ecosystems are going to play a huge role in us mitigating climate change, making sure that we still have the ecosystem services that we need out of these very productive forests. So I just want to give you an idea of sort of overall how I get at asking this question. So like Sergio said, my lab kind of has three separate parts. I look at the patterns of plant distributions, both using sort of online databases and doing field work myself. I think about how that fits into our understanding of ecological theory. And I'm especially excited about sort of thinking about plants that maybe we didn't build our theories off of. So these non-vascular plants or these herbaceous plants or these smaller plants rather than trees. And then the third part, as Sergio mentioned, is thinking a little bit about how we manage our forests for diversity. And all of this is wrapped up into that central question of understanding the drivers of diversity. Today, I'm going to tell you just about a couple of projects, but I do a lot of work that is both experimental and observational. So these are some of the tools that I use across my lab and that I've been starting to set up after just starting at Lehigh as a faculty member just a semester and a half ago. And I do global work, so mostly in the Americas, so North America, Central America, and South America. And that is what I'll tell you about today. So just to give you an idea of where we're going, before I tell you about my research projects, I do want to give you a bit of motivation. So why do I study community assembly in these small plants rather than in trees? Then my first project I'll tell you about is a new project. It's sort of an unpublished work where we're talking about epiphyte niche differentiation. The second one is also going to be a new project that integrates this niche differentiation with epiphyte enemy escape that I just published late last year. And then I'm going to end with a final project thinking about species interactions among epiphytes themselves. So that's where we're going. And I want to just get everyone on the same page about what do, Sergio and I mean when we talk about forest community assembly. So this really is the process of arrival of propagules to a new site, whether it's, primary succession, where it's a totally new site, or you've had a disturbance and now there's an opportunity for new propagules. We then sort of track the development of these plants through time, watch them grow up to maturity, and then we see sort of what species drop out of the community and which come back into it, right? So the development and the changing of the species turnover over time. And then finally, this thought about what maintains diversity. How do we have such high biodiversity all in one spot for perpetuity? The idea here is that if you can understand this process, going from arrival to establishment to diversity, then we can understand and make sure that we're managing for these high diversity ecosystems. On the other hand, if something goes wrong and we have a really low diversity area, we can try to sort of get that back on track and have successful assembly and understand what drives high diversity. Like I mentioned, so when I would do work in forests, when you think of a forest, probably the first thing you think about is trees. Absolutely most of the carbon in the forest that's standing there. But what I want to give you just a bit of data to show you is that we're also missing quite a lot if we're just looking at the trees. So here's just a few examples of the many other types of plants that are a big part of that forest as well. And this was something that actually early in my PhD, I got really excited about and thought this was a very easily answerable question that must have already been done. So I just wanted to know which of these major plant growth forms, trees, shrubs, lianas or woody vines, herbs, and epiphytes are the most speciose. How many species are there of each of these groups across the world in different forests? But it turns out that was not answered yet. So I looked up data from 18 different forests all across the Americas, from North America to South America. And I basically looked at the relative proportion in these temperate and tropical forests of all these different growth forms. Now, we of course know tropical forests are more diverse overall, have more species. So this is relative species richness, the proportion. The first thing that came out of that is that trees are the minority in both forests when you look at relative species richness. So in the tropics, 30% approximately in the American tropics, it's about 30% of trees. And then in the temperate zone, it's only about 7% of all of our vascular plant species are trees. In the tropics, we saw this sort of more even distribution across the different growth forms versus in the temperate zone, like where I'm in Pennsylvania right now, herbs make up about 80%. So this is really the driving force of what I did for sort of why I study what I study. We formalize this into what we call the forest physiognomy concept. It basically just says where you see diversity in these different types of forests differs. It's more upshifted in the tropics, downshifted in the temperate zone. And we also know these plant growth forms are going to be different across these different types of plants, of forests. So to sort of wrap up, why do I do what I do? We know that non-trees are really vital to forest diversity. So if we want to understand mechanisms of diversity maintenance, we should probably look at the majority of the forest or other forms of plants, not just the trees. In my lab, I have a new PhD student who is working on invasive species to really look at this in the temperate zone. And I've really started looking at these growth forms in various ways. We just got a paper submitted or accepted that we also are looking at sort of how these herbaceous plants in the temperate zone, because they're the majority of the diversity, are sort of changing over time and how phenology plant changes with climate change. Oops. And I also have just participated in a herb diversity workshop last spring that we're currently writing up. So this is a really cool group of international people that we are looking at exactly how we can bring in these herbaceous plants, these non-trees, into our forest geoplots, into our surveys across the world. So that's sort of where we're headed with thinking about the sort of smaller plants and their communities. So I want to leap in. I have, like I said, 3 research projects I want to just introduce you to today. And this first one is really a labor of love that I've been working on for quite some time. So starting out with epiphyte niche differentiation. Now, most of you probably have seen epiphytes in the wild right around your university. However, in case you haven't or haven't thought much about them, these amazing plants are basically non-parasitic plants that grow on other plants. So they are not directly taking nutrients or water from the tree they live in, but they do need those trees to access the canopy, get to the light. And they're really vital parts and very, in my opinion, charismatic plants, both in temperate and tropical zones. So they make up about 20% of neotropical diversity and 9% of vascular species across the whole world. And in temperate forests, such as these ones that are temperate rainforests out in Western US, for example, or in Canada, they also make up more than 4 times as much foliar biomass as the host tree itself. So they're really accessible nutrients for a lot of other organisms in the ecosystems. Now, I want to walk you all through niche differentiation. This is just to make sure we're all on the same page about the process I'm talking about. So if, of course, if we're just looking at a sample of walking out into a forest, and you have no variation, let's say, of one important resource, so your water environment, this one species who is in its perfect environment is going to be dominant everywhere. However, as soon as you have a gradient where there's maybe wetter soils on one side of the forest and drier soils on the other, you're going to be able to have variation in who is more efficient at collecting water in those different environments. So you can see if there's this second species that is really good at surviving and being competitive under low water environments, that species is basically going to outcompete the first. So then we're going to have 2 species coexisting. Now, let's say we have another gradient going the opposite direction, right? Maybe this is fungal pathogens. Well, if we have few fungal pathogens up here, many fungal pathogens down there, then we also have this opportunity, this niche that we can sort of vary and have additional species. So if we have these different types of species that have varying degrees of being good at defending against pathogens, for example, versus uptaking water, then you're going to have no species that's good at everything and coexistence of all of these different species. So this is basically the idea of how niche differentiation works. No one species is perfect at everything, and so you have these trade-offs. Now, my big question is, how might these epiphytes, these plants that grow up in the canopy, assemble differently than what we look at the forest floor? And to think about that, we want to move this whole sort of idea of niche differentiation into 3 dimensions. So there's actually an extremely strong gradient when you go from the understory up to the canopy as well. So abiotically, we know it's humid, cool, and shady in the understory, hot, dry, and very sunny in the overstory. We also have some evidence, which I'll talk about in the second project, about a biotic gradient as well, of the microbial community being much more diverse and abundant in the terrestrial soil than in the canopy soil. In addition, because we're actually talking about live host trees, we have these vertical substrates, these substrates that are the trunks versus horizontal branch substrates. This is what I basically call the 3D niche differentiation hypothesis. We just know that there's these spaces in 3 dimensions as well as you move up from the ground to the canopy. So I would propose that these epiphytes are really adapted for these specific microsites along these gradients. You can envision perhaps this orchid species does really well in that hot, high, dry area versus this aroid does a lot better in that cool and shaded, humid area. Likewise, maybe these ferns are particularly good at grasping onto a trunk substrate versus this bromeliad needs a really nice, thick horizontal substrate, a branch to be able to establish. Sort of in line with some theory that we know about elevational gradients, you might predict that there's actually a mid-canopy peak, just like a mid-elevational peak, where you see this overlapping of a lot of different microsites all at this one spot. So I went out to test this in 2 different field sites. One I set up at the end of my PhD down in Santa Fe National Park, which is in central Panama. And then in my postdoc, I set up this other site in 2020 in the Hoh Rainforest in Washington State. Now, these are both very diverse forests with a lot of epiphytes. This is the temperate rainforest of Washington, and this is the tropical cloud forest of Santa Fe National Park. Of course, we do canopy work, so we are actually climbing up into the canopy and observing the plants where they are. And this is absolutely teamwork. So I just want to highlight a few of the many students that I've been able to work with, undergraduates, recent graduates from the US as well as from Panama, where we basically go up and we're able to sort of enjoy all of these plants exactly where they are. So I really need to shout out to these many field assistants and students that I've been able to train and work with over the years, since you cannot climb alone, and it's been such an exciting area of research. So the way that we set this up, I'm going to first just show you some results from observational studies. We went up into the canopy, 6 different survey sites in each tree, kind of nested across these 3 height zones, so low, mid, and high in the canopy. These are 20 by 20 centimeter plots. These may sound small, but in just one 20 by 20 centimeter in Panama, we can actually get up to 30 different vascular epiphytes in this one small spot. So this is a picture where you can see we have 12 different vascular epiphytes in this 20 by 20 centimeter plot. That's kind of why we're using this size. In some plots, we have one giant bromeliad that takes up the whole plot. In other plots, we have 35 little vascular epiphytes. So just kind of depends. We did half of those on the trunks and then half of them on the horizontal branches. Now, we did do fewer in Washington just because there's a lower diversity overall. It's mostly non-vascular epiphytes in Washington. So we have 12 trees we climb every year in Washington and 30 in Panama. So what did we see? Just across the bottom, we're going to look at these different microsites. So here on the left is the branches, on the right is the trunks. I'm going to show you just species richness today, but we have a lot of other diversity responses as well. Within these different groups, we have low on the left and high on the right. So we're going from the lowest branch to the highest branch, or the lowest spot on the trunk to the highest spot on the trunk. The first thing we found in the temperate forest is that species richness was higher on the branches than on the trunks. However, that was not true on the lowest vertical plot. So this is basically an interaction that we see. The vertical substrates have a slightly different pattern than the horizontal substrates. In the tropical forest, instead, we saw the same pattern where we see the branches being higher overall than the trunks, right? But we don't see that sort of the lowest trunk looking different. Instead, what we end up seeing is that across the whole board, we have much higher in the branches than on the trunks. So what's going on? One of the things that I want to just give you a visual for is that when you look at a temperate forest, that's a rainforest, you have this really thick mat of bryophytes all along the ground. And what we're seeing is that that bryophyte mat is actually climbing up and starting to be somewhat epiphytic, pretty low on these trees. That does not happen in the tropical forest. So this is a picture of Santa Fe, right in the midst of field season. You can see it's pretty muddy. And so you don't have that thick bryophyte mat on the floor that only occurs on the trees. So this is one of the differences that we think is actually explaining that sort of slightly higher diversity than we expected in the low trunks or the low trunk strata in the temperate zone versus what we see in the tropical zone. So to bring it back to our hypothesis, it looks like in the tropical zone, we actually did see this sort of higher biodiversity on the trunks and sort of up a little bit higher, sort of where we expected, versus in the temperate zone, we actually saw a higher biodiversity down in that V1, which maybe overlapped with that ground layer of really thick bryophytes. And then we also got some more epiphytes up on those branches. Now, I told you I do a lot of experimental work, so I just want to highlight sort of what we've been continuing to work on is actually experimental work as well. So that was the observational work in each of these plots all across Panama and Washington. We've actually also done removals. So here is just an example of where in 2017, I removed 20 by 20 centimeter bryophyte mats with all their epiphytes, and I'm comparing that to a control plot. So I reported on the control plots today, but we also are looking at secondary succession. Now, just to give you a visual of what this looks like, so I don't have a picture from 2024, even though we did just go back in January, but you can see this sort of pretty rapid secondary succession. And this is really novel. There aren't that many studies that do this experimental work. And you can see that by 2022, just 2 years ago, we have almost complete coverage of bryophytes, and you have this large clump of orchids on the side and the piperaceae right here. So this is really exciting results that we're working on publishing now. Like I said, we just got back from the field. This is my field team from January of 2024, my first field season here at Lehigh. And so I just want to, again, shout out to my amazing long-term collaborator, Josue Ortega, and then also my field team from this year. And again, most of these are students from the University of Panama right in the city, and also in Santiago, which is the second largest city in Panama. Okay, so we talked a little bit about niche differentiation. Now I want to introduce you to this second hypothesis. This is one that, again, we just published this study a few months ago at the end of 2023, and we're looking at how potentially epiphytes could also be escaping from enemies. Now, the origin of why I was interested in this is because epiphytes, like I said, have a few differences between terrestrial plants. And so one thing that we notice about trees versus epiphytes is how they recruit and what their community patterns are. So one of the well-established patterns we know from tropical tree seedlings is this pattern of negative density dependence. If we have one species of tree, this green species, what we see is a very strong pattern where you have a low likelihood of survival of that same species right underneath that mother tree, and the higher survival of juveniles is going to be away from the tree, right? So it's negative density dependence. When you're right underneath that, the area of that mother tree, you have low abundance. Now that means you have the opportunity for a second species or multiple species to recruit underneath it. And we know that now actually soil microbes play some role in that pattern. And so we know that for tropical trees, the interactions between soil microbes and juvenile epiphytes or juvenile trees and the way that they recruit is a major mechanism for diversity maintenance. Do we see the same pattern in epiphytes? No. So one of the things that's interesting with epiphytes is that when you are in one tree and you're just looking at the distribution of species, a lot of times you're instead going to see this clumped distribution. So you might have one large adult, and then you have a ton of little juveniles right around it. So this is the opposite pattern where we basically see intraspecific potential positive density dependence. So this is sort of the opposite pattern of what we see in trees. Now, of course, there's a lot of things going on, including things like dispersal. But one of the things that we also know as we zoom out a little bit is that that also happens sort of on the larger scale. So we have one tree in the middle, let's say, that has a ton of epiphytes. It's this big old tree with 400 different species of epiphytes in just one tree. Well, that has a positive impact on the trees around it as well. So we actually see this sort of clumping patterns of diversity and sort of abundance within trees and across trees. So that is different. It's just a different pattern than what we see in terrestrial ground-based trees. And so what I sort of have started thinking about is, well, what if, if we know soil microbes are something so important for keeping trees away from each other if they're the same species, what if these epiphytes aren't experiencing that, right? Maybe they don't need to be defended against these soil microbes anymore when they are up in the canopy. So this is exactly the core of this hypothesis that I've been working on recently, the epiphyte enemy escape hypothesis. So sort of like how we talk about species that are outside of their native range, being able to escape enemies such as herbivores or even microbes, perhaps these epiphytes, by moving up into the canopy, are escaping some of these soil enemies that are so detrimental to terrestrial trees. So this is what I really want to explore in this project, is thinking a little bit about how these terrestrial soil microbes might potentially be contributing to these patterns as well. Now, there is one other study that I was really excited about when they published it in 2019. So Evan Gora published this study that basically looked at what types of microbes lived in the canopy strata. So if you haven't looked at if you're not used to looking at community matrices, all you want to look at in these next two figures I'm going to show you from their paper is that the closer together the circles are, that means they're similar communities. The farther apart means they're dissimilar. So what they found when they looked at soils that are and microbes that are sort of on rotting branches, as you move from the forest floor to the overstory, so you have this black boxes being the forest floor, and then you move up through the canopy here, is that there's a huge difference between terrestrial microbial communities and arboreal microbial communities. There is also a gradient moving up the tree, but the strongest differences are soils on the ground versus soils in the air. This is true for bacteria, and it was also true for fungi. So the fungi are even more strongly differentiated. Terrestrial soils, very, very different. So what I was able to do is take advantage of fallen epiphytes that have actually fallen on their host tree, to be able to really test this. Now, I did actually combine, so I'm looking at this epiphyte enemy escape hypothesis with the same hypothesis we just talked about, that niche differentiation, because we know that where the epiphyte came from is also very important. So what I did is I basically went to 4 different donor trees, measured where those epiphytes, these fallen trees, where the epiphytes were on the original host tree, and then I collected them to transplant them to a new tree. But the way I transplanted them was so that they're either in contact with the soil right at the base of the tree, so they're actually touching terrestrial soil, or just a little bit up at 1 meter or at 2 meters. I put these 3 epiphytes in 90 different recipient trees, and then we basically monitored them, so monitored their health, how they're doing for a year and a half. So the first thing, just looking at whether or not they performed worse, we did find that there was a slight difference between those ground epiphytes and those epiphytes that were transplanted just off the ground. So again, we have very small amounts here of differences. It's just a meter or 2. But we do see that it's basically a 50% survival rate versus a 66% survival rate. So when it comes down to it, that does make quite a difference. It's just a year and a half that you have that difference in survival. We also have health status. So we were looking within these different groups. We also have the lowest health status. So this is sort of like a indicator that I used from other epiphyte studies, where you have lower health status as well in the epiphytes that are touching the ground in comparison to ones that are just off the ground. We also quantified how much leaf loss they had over time, and it was a very similar pattern where we basically had the ground epiphytes, the one touching the terrestrial soil, lost more leaves overall than the epiphytes that were just off the ground. We did also look at where they came from. So I'm just going to briefly show you a little bit of that data as well. Here we're looking at survival probability and where it comes from as colors. So these are now going to be tracking through time. The last slide I just showed you was just at the end of the experiment, what was the total? And you can see these are ordered exactly how we expected, where these epiphytes did the best if they came from low in the strata, versus when they came from the upper strata, they died off a little bit faster. So that's sort of thinking about where you come from matters with how, well adapted you are for being in contact with the soil. Now, we also looked at across different taxonomic groups because we weren't specifically looking at any one taxonomic group. We had sort of 5 major taxonomic groups. And we did see differences there too, where aroids, for example, did do the best. And then that in comparison to orchids had the steepest drop off for their survival, which is also something that wasn't super surprising, but hadn't actually been well documented before. So why be an epiphyte? This sort of gets at it. One of the sort of cool results of this then is that it looks like there is both a role for this vertical niche differentiation that we talked about in the last project, as well as potentially some of these soil microbe interactions that may be contributing to why it might be worth it to have all those adaptations to be living in the canopy. So it looks like being an epiphyte on the forest floor is not good, and you will die faster if you have contact with that terrestrial soil. So for the future, one of the things I'm really excited about is actually digging into that a little bit more and directly testing the microbial epiphyte community interactions. And so this is actually something I'm working on, applying for lots of funding right now and starting to collect some preliminary data to really get at what that microbial community in the canopy looks like. I also just put a couple of other projects to kind of give you an idea of what some of the cool things that we have been able to incorporate into this big experiment. If you're going to climb a tree, collect as much data as possible. So we actually have some canopy traps up in the canopy to look at plant-animal interactions. We just started collecting microbial communities, and we also have abiotic monitors, iButtons, that are up there. Just one more cool picture from this particular collection. So just 2 summers ago, I worked with a Yale undergraduate. She was doing her senior thesis with me, and we basically collected microbial communities, fungal communities from the canopy, going all the way up this gradient in Washington. And we just sequenced them last year, and so we're writing this up into a paper now, thinking about how the fungal communities change as you go from low in the forest to high in the forest, and how they differ across different host species of epiphyte. So stay tuned for that. She also was actually able, so she did her thesis with me, but now she actually started as my lab tech. So we're still collaborating, and she was one of the people who went down to Panama with me this past January to climb the trees as well. So now she's been doing temperate and tropical epiphyte work. So for my last project, I want to just give you, in the last 10, 15 minutes we have together, is to think directly about these species interactions within the epiphytes. And this is something that, again, is just a really exciting area that I'm super excited to be starting out. This 4th project is not published yet, but we're starting to work on it and writing it up now. So we know overall biotic interactions are ubiquitous. There's tons of different ways that plants and animals and microbes all interact. But one of the things that we didn't know, and that I wrote sort of a review paper about early on in my postdoc, was actually, what do we know about biotic interactions in epiphytes? Because they're so strongly associated with these abiotic environments, right? Like hot and dry canopies, and they have all these cool adaptations. There really isn't that much literature about biotic interactions. So I did a review paper early in my postdoc to just answer this very basic question. This is the one that I'm going to talk a little bit more about with you, is this epiphyte interactions. It turns out we know quite a lot about epiphyte host interactions. There's over 300 published papers on epiphyte host interactions. We know quite a lot about vascular epiphytes and animal interactions with pollinators. But these 2 on the bottom are just really, we don't know much at all about them. So I'm going to tell you about a small project that I did in my first couple of years of my postdoc that are looking exactly at epiphyte interactions. So just to give you an idea of like how little we know about this, I'm going to focus on non-vascular epiphytes for this project, since that's where I was out in Washington. There's more non-vascular epiphytes there. You can see on number of publications, there were only 5 publications on facilitation and 8 publications total on competition in these non-vascular epiphytes. So huge open gap in the literature of trying to figure out who's interacting with who and how. And again, this is in Washington, so these are these non-vascular epiphytes. So I had kind of just to ask the extremely basic question of how are epiphytes interacting with each other. I had a couple of predictions. So this is sort of based on stress theory or density theory, where if you think about when an epiphyte would benefit from another epiphyte being there, we would expect that if you have really low densities, especially in these pretty harsh environments early on, epiphytes might actually be buffering these harsh conditions, and so they would be facilitating other epiphytes. So positive interactions when you have low density. Now, these are 2 examples that are just pictures I took out in Washington, where you can see an early sort of early accumulation of epiphytes, where you have this bryophyte that's moving in on this small piece of bark here. Here's another great example of a new branch that has a tiny little clump of mosses that's right in the corner of one of those branch bifurcations. So we would expect this where facilitation would happen more so at low density. The other side of that prediction is that, well, at high density, when you have these super thick epiphyte mats, these epiphytes are probably competing for space. So this is another couple of photos. Like I said, in Washington, these epiphytes can get really thick. Same in Panama, we actually have, sometimes we can have a bryophyte mat that's almost half a meter thick. And so you're really seeing a thick, lots of layers of epiphytes. So what you can see here on the upper left of this picture, this is one species of moss that is basically encroaching and growing over a second species of moss that's right here. And then the same sort of on the right side, you can actually see that these epiphytic bryophytes are sort of all intertwined with this epiphytic lichen that are all interacting in the same exact space. So my prediction would be at high densities, these epiphytes are competing rather than facilitating each other. Okay, so I put together this basically mesocosm experiment. So I collected epiphytes from the field and then put them into sort of small containers and then grew them up in sort of like a outdoor greenhouse area. We use this density treatment approach to specifically get at that density question. So instead of the more traditional approach where you say, with one species with a self versus one species with another species, which does it do better? We had that also as sort of a backup, but our main project was actually looking at this gradient of densities with 7 different species all at once. So we're going from low density to high density with all 7 species. We had 5 replicates for all of our treatments, which means we had 90 total mesocosms. Each of those 90 mesocosms had 7 species in them, and we painstakingly weighed them before we did the experiment, set them out, and then weighed them again. And so again, I have to shout out to one of my amazing undergraduates at the University of Puget Sound, Beatrice Bugos, who helped me weigh all of these tiny plants over and over again for months. So what did we find? So across the bottom, this x-axis, we're just looking at epiphyte density. So again, going from few interactions to many interactions, just to give you an idea. So this is performance, and we did track this like per species, per individual, things like that. So they are going to be kind of normalized here. Again, you would expect a positive line here if we were looking at facilitation, and a negative line if we're looking at competition. So what did we actually see? So this is all epiphytes together, all of those 7 species times 90 mesocosms. And what you can see on their relative growth is that there's actually quite the interesting curve. Now, overall, clearly positive, right? So we have a positive slope across this entire range of, really low density. So those are our far apart epiphytes to really high up here. So sort of all overlapping. However, it's really interesting because what we do see is that there's this sort of tapering off. So you have much steeper slope here in the sort of low density than what you have over here in the higher density area. We also divided this out across different species. We know some of these species are sort of early, mid, and late successional species. And so we looked at this, and you can again see the sort of difference where you have where you have across the species. Didn't matter as much which species we're talking about. Early sort of initial mass being low, so those are the low density ones, very steep slope, and then it tapers off. And in some of these species, it does actually taper off to be pretty like even. Now this is, so overall, we still have this strong facilitation that does have this tapering off. So that is basically right in line with what we would expect from our stress gradient hypothesis. Interestingly, we specifically based this on a different study that was done in a greenhouse, but with terrestrial bryophytes, some in the same genera. And they concluded the exact opposite. So one of the things that was really, really cool as a result of this is that for epiphytic bryophytes, we're seeing facilitation that tapers off a little bit to less strong facilitation. In their experiment, in which they used pretty much the same protocol, but terrestrial bryophytes, they concluded that they're all competing because they looked at these and said, hey, this is a negative relationship. So one of the things that's really cool to think about is whether or not terrestrial bryophytes are behaving and fundamentally have different overall interactions of competitiveness than these epiphytes that are the same, genera, except they're living up in the canopy. So that was a really cool thing that we got out of this. And it does make sense that if you're in this harsher environment, then you'd shift more towards that positive space, that facilitation. So this is a really exciting initial result. Again, I just want to sort of give you a little idea of where we're going next with this. We actually also have a cool facilitation experiment in Panama and in Washington. So in addition to doing my removal experiment, which I already told you about, we actually also did a facilitation experiment. So in some of these, we actually, in Panama, what we're interested in, do these bryophytes facilitate vascular epiphytes? So if you look at this third plot here, this is the original where you can see these orchids and pipers over here. And here I've removed them, but I left the bryophytic epiphytes. So by comparing these two, I'm actually going to be able to show whether or not bryophytes are facilitating the vascular epiphytes in their reassembly. So this is a really exciting direction that we're working on. This is what that looks like in Washington. Because they're mostly bryophytes, we actually added a facilitator instead. So we're going to be comparing sort of how does a totally empty plot compare to an empty plot that then has a known facilitator species as a tiny little propagule to start out reassembly. And that particular project, so the one in Washington, my current master's student, Vanessa, is going to be going out there this summer and resurveying all these plots and figuring out whether or not we do see facilitation across these different species. So with that, I'm going to just give you a few take-home points. First of all, these canopy plant communities are, in fact, assembling along these sort of distinct vertically stratified niches. So that was that first project that I told you about. Second, I told you a little bit about terrestrial soil microbes. It looks like from that second project I told you about, they're particularly bad for these canopy plants. And I'm really excited to get into the microbial community to figure out who are they, what are they doing, how are they interacting with these plants. And then the third project, it looks like these bryophytic epiphytes are more positive interactions than the terrestrial counterparts. So being an epiphyte means you might have more positive interactions with other epiphytes than if you were a terrestrial plant in the same genus. And all in all of this kind of brings that initial point home that non-trees are essential components of forest function, diversity, and also extremely interesting to study. So with that, I'll acknowledge, again, there's tons of people who've helped me do all this field work, my collaborators here in the US, as well as in Washington, or as well as down in Panama, all of the land, and especially all of the funding as well that I got to do all of the work I presented today. So with that, thank you very much. I think we are just in time to have some questions.
Absolutely. Thank you so much, Michelle. I always love your presentations, and I'm really thankful that you spend some time to talk us about the different projects and how have they evolved through time, which is I'm super excited to see how the results are coming together. Um, okay, so we have plenty of time for questions, and I'm going to start with two questions. The first question is, you showed us a lot of pictures of students working with you in the field and working up in the canopy with people is how you do it with people, ropes, and what so on. I'm curious now on how do you usually translate this conceptual questions and these theoretical questions about assembly or reassembly or succession, right? Or competition versus facilitation? How do you, like, what language do you use, or how do you educate your students and tell them, look, what we're thinking about here is X, Y, and Z, instead of just going all the way technical? How do you kind of break it down and tell them this so they can start building their own capacity to understanding the process?
Yeah, that's a great question. And I would say there's sort of two things. So currently I'm teaching a course of non-majors that's just introducing people to regional biodiversity. And it's 45 different students, and they are all learning about biodiversity pretty much for the first time. And a lot of these concepts I do sort of break down into sort of like how I showed you with, niche differentiation or community assembly of like showing like what are the steps and what is the broad concept. So I do use a lot of animations. I use a lot of visuals in all of my lectures when I'm teaching students, especially if they don't have much experience with it. And the second thing I do is very participatory. So even though I have 45 students who are non-majors, I have business students and health sciences students and engineers. We actually go outside and collect data. So I mentioned I use camera traps in Panama. I actually also have camera traps here that we actually put out camera traps, and then the students are looking at the photos in class, counting what plants or what animals we're seeing, and then we're actually quantifying those and putting them together as a class to see, like, to test hypotheses. So, does animal occurrence correlate with human occurrences, right? Like, are we seeing trade-offs between human activity and animal wildlife activity? And these are things that these students may never have thought about, how to quantify biodiversity or animal behavior before, but by counting them and then putting them all together, like, they're doing it. So I think that's the other thing that I try to do is put together actual experiences where students are collecting data and testing hypotheses themselves, at the same time as like really breaking it down to the basics.
That sounds really amazing. Thank you. Yeah. Um, yeah, and it makes total sense to involve people in the process to have some buy-in in their own, like in their own learning process, right? It's just not like a sponge receiving, but also we can do this together as a group and collect data. That sounds really awesome. My second question goes back to epiphytes. And so I remember years ago, Gerhard Solz published a paper talking about how um, that he thought that the occurrence of epiphytes on trees was mostly a neutral random process, right? I think maybe he came from that idea of, neutrality and Steve Hubble and whatnot, and he was kind of like on that bandwagon. And now you're talking about niche differentiation, but how much this neutrality or randomness is taking place in the arrival process of the community assembly?
Yes, so this is a great question. And there is actually still a lot of debate about when and how epiphytes assemble, whether they're more neutral in some areas or more deterministic in other areas. One of the things that I do, he has a lot of amazing work from BCI and from the San Lorenzo plots. And so on BCI, that's a seasonally dry forest, it's lowland, and the coverage of epiphytes is going to be different. It's a lot lower than what I see in Santa Fe. Now, that being said, certainly there are a ton of these arrival processes that are sort of like, random. And there's been a couple of good papers about sort of thinking about epiphytes as meta communities too. And one of the things that's very clear is that in a lot of systems, epiphyte communities are not at capacity. So you're continuing to assemble. You're not, at a peak community, you aren't full. And so that's something that I think there is a lot of that discussion of, these are dynamic ecosystems, a branch breaks and the epiphyte falls to the ground. So they're very dynamic. So there are a lot of cases where you're basically just like, whoever gets to that spot is going to get a good spot and then start going. So there absolutely is a lot of stochasticity there. But there's also, there's other people like Carrie Woods, who is my first postdoc advisor. She literally has also published a paper about this, like, basically deterministic turnover in Costa Rica of this turnover within trees. So I think there's actually room for both. There's arrival processes and some certainly stochastic processes that we just don't have that many. Like, we don't have nearly the trait data that we do for epiphytes as we do for trees. And there's so many big open questions. And like I said, basically like any sort of biotic interaction of like, well, are epiphytes truly like, succession, are they going through a successional trajectory, and like what species replace the others? Why are they doing that? Tons of open questions still, just about basic succession in epiphytes.
Thank you, Michelle. So there is one question in the chat, and then I'm gonna give the microphone to Juan. The question is from Paula Morales, and she says, another question, do you have projects running with grants available waiting for PhD students? So maybe Paula is interested in joining your lab. So I don't know what you have to say about that.
Pero antes, Paula también hizo una otra pregunta. I want to know
sí,
what
perdón,
will be...
no la vi.
Sí, okay.
The interaction, yes. So I'm looking at this one.
Okay, thank you.
So Paula also says, I want to know what would be a possible hypothesis for the interaction
between vascular,
during colonization of trees. And this is definitely a really interesting part that I'm not so, Paula, are you thinking between like vascular or non-vascular?
Yes.
Yes,
vascular.
Yes. So this is one of the parts that's a little bit, again, not well studied. So I don't know of any tests that directly look at across succession vascular interactions. What I'm testing with my my project I talked with about as like a future direction is the bryophytes facilitating the vascular epiphytes. And there's a lot of reason that we might think that would be the case, right? They're these sort of extra humid microsites. They have a lot of texture that the seeds of the vascular epiphytes could sort of colonize, arrive, and then stick into. But they're definitely, for Washington, we do see that sort of preliminary interaction that's positive, right? Like the more epiphytes you have together, the better they do. And I don't know of any direct tests yet of the vascular epiphytes. You would have to test that in a place that has really high density of vascular epiphytes. So of course, if you have, let's say on BCI in central Panama in a lowland forest, if you have one vascular epiphyte here and another vascular epiphyte all the way over here, the interaction is probably not significant, right? So you'd want to test that in a place where you do have really closely put together vascular epiphytes. And it could be, there are some like just anecdotal, like I see epiphytes that are, maybe a big tank bromeliad, and because it's accumulating all of this humus, it's accumulating leaves, you actually have other small epiphytes that are around it, in it, on top of it, right next to it, that are different species. So things like that could potentially be facilitation, but it hasn't really been studied very, very strongly.
Thank you, Michelle, and thank you, Paola. Sorry, I missed your first question, Paola.
And the
okay.
follow-up question, so I am currently applying to grants to get funding for PhD students. Neither of my students currently are grant funded. So I will put on my website if I get a grant that would support a graduate student. And I think, you know, certainly I would be very excited to recruit graduate students from Latin America who would be interested in doing PhDs. Because it is absolutely, so like Sergio said, I also speak Spanish, and that's totally necessary for working in Santa Fe. We are out in the rural area, and so I think it would be a great, you know, a great fit to have students who can be used to being out in tropical forests and, you know, speak with all of my collaborators in Spanish, and, you know, feel free to keep looking at my website, but I don't currently have any funding for this upcoming year, and I'm applying for lots of grants to try to get some.
Thank you, Michelle. All right, last question from Juan.
Yes. Hi, Michelle. Thank you for this great talk. I really enjoyed it. Great way of summarizing tons of data. I actually want to ask you a question which you've probably heard before, and you were kind of touching on, which has to do with a trait-based approach, more functional ecology. Have you explored a little bit more like how traits change both vertically and within the community assembly process with these different filters, both biotic and abiotics you have in the different parts of a canopy? Because I think it's really interesting. You probably have very different ecological strategies within an area, and maybe a lot of variation within a category of ecological strategies. So it's a very complex system that I think could benefit quite a bit from a trait-based approach. So
yeah. So
what are your thoughts?
Yes, I definitely think traits would be a great direction to go. And it is both a part of what my master's student is actually going to be doing this summer in Washington. She's going to be collecting traits. We don't even have traits on like bryophytic epiphytes at all right now. So she's going to be collecting bryophytic epiphyte traits, and exactly connecting that to like, as you move up in the canopy, are you seeing this turnover of traits that would be predicted by succession? So absolutely, that's something that we're starting in Washington, and is a part of one of my aims for the proposals I'm writing for Panama. Is we need to understand more about the traits that underlie these sort of, like, I think we're going to see potential trade-offs between defenses, right, and tolerance to being up high in the canopy. And so those are all things that are traits that approach has not been taken really yet. And that's something we do have some good information about, you know, sort of basic traits like photosynthetic pathway, right? Like, are they doing CAM photosynthesis? There's some really great work on epiphytes and CAM photosynthesis. But we're really quite sort of data limited still. And there are some new, so Gerhard Satz and some of his mentees have put this new sort of trait map together, and there is now some good epiphyte trait data out there. And so I think we're right at the right moment to start testing hypotheses about where do we see these traits, how do we see them turn over, because now there's just at the point where we're getting some good trait data that's publicly available and part of these sort of data networks.
Great, thanks. Yeah, I look forward to see some of those results.
Yes, me too.
Yeah. Thanks.
Well, thank you, Michelle. Thank you, Juan, for that really cool question. And I want to thank everyone that joined us today for this fantastic talk. Please stay tuned for our talk next week as well. Really interesting talk about turberas in Orinoco and Amazonia. Michelle, thank you for enlightening us about niche differentiation in epiphytes. Thank you for explaining everything so clearly. I'm always amazed about your capacity to break things down and explain it so thoroughly. Thank you again, and I wish you a very happy Friday and a very great weekend. Thank you, everyone, for attending, and have a great day.
Ciao.
Ciao. Gracias.
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 Bueno, muchas gracias.
Gracias. Hasta luego.